Thanks, Colleen. Thanks, Stephen, and thanks to all the organizers for inviting me to speak today. It's an honor and a pleasure. Thank you all for coming. So today I have a relatively short amount of time to cover the last 20 or so years of research. The basis of the research really began in the early to mid-1990s about from genetic labs like my own, where we were studying simple organisms and trying to understand why they grew old and why didn't they live forever. And we've now, after the last 2 decades, come to a point where we believe we found the key regulators of aging in mammals, and these genes control most of the life course that we follow as human beings, including dementias. And so that's what I want to tell you about today. So we already know that some people are genetically predisposed to live a long time. These are so-called Centenarian families. This is a father and son. You can see 70 and 100 looking pretty good. These are from New York. And my buddy near Barzilai has been studying Centenarian families for many years and has many samples from these individuals, and he's been characterizing their genes and picking out certain genes that allow families typically to pass on longevity in their family. And he uses the in-laws as controls. Yeah, the negative controls. Anyway, so one of the things that we've seen over the last, I would say, 200 years at least, is that though we've greatly improved our ability to live longer, we haven't always improved our ability in the last 5 to 10 years of life to live better. It's actually what's happening is that because we're targeting one disease at a time with one medicine, some aspects, typically the most difficult is brain degeneration, dementias, that we end up with people who are living much longer, of course. Cardiovascular system is protected, but their brains are not. And so actually, to put this in a graph, this is from the UK, the percentage of our lives that are spent in good health is actually declining, even though we're living longer, which is really something that we want to prevent and reverse with the research that I'm going to tell you about. So I work on aging, and what the typical question that I get from scientists and lay people is, why would you want to live older in an old age? Why would I want to be older in a nursing home for longer? And the argument there that I give back is that we're not really talking about being in a nursing home for longer. We're talking about keeping people out of nursing homes for longer and extending what we call the health span, not just the lifespan. And in fact, from the studies I'll show you in mice, I don't know how to extend lifespan without improving the health of the animals. And another reason for doing this research is that this is from the RAND Institute, RAND Corporation in the US. And Dana Goldman is an economist, mathematician, who has looked at what the various costs are to extend an additional year of someone's life. And this includes the research and development as well. But you can see pacemakers and anti-angiogenesis compounds and molecules are extremely expensive. Compare that with something that at least is theoretical, but we're getting closer towards it. The FDA thinks that this is coming down the pipeline very soon in the US. The anti-aging molecule, for want of a better term, could extend lifespan for a mere $9,000, and that includes the research involved as well. I won't show you the statistics, but there are numbers that say that even in just one country, the US, the savings would be in more than $3 trillion per year. Okay, so hopefully I've convinced you this is something worth working on. I was interviewed by somebody this morning, ABC, and they asked me, well, why would anybody want to live longer, and isn't aging something that's natural? And my argument there is that cancer is natural, Alzheimer's is natural, diseases are a natural course of life. So why is aging any different? Really, all we're talking about here is the difference is that instead of having one pill for one disease, we have one pill for a dozen diseases. So I studied at UNSW. I'm still very much an Aussie, though I have one lab at Harvard University. I headed off to MIT in Boston to do my postdoc while my mother was recovering from surgery to remove this tumor in her lung, and this is actually very similar to the tumor that my mother had. And that drove me to try to figure out ways to actually prevent and treat diseases of aging, including cancer and Alzheimer's. So this is a little bit of a story just to lead up to where we are now. This is my mentor at MIT when I was a kid. And what we decided to do was to study yeast cells. And this is a representation of a yeast cell, the organism that you owe your beer and your wine and your bread to. And we were trying to figure out, why does a yeast cell die after just a week? And what we discovered was one of the major causes of aging in the yeast was the change in the packing of the nucleus. So these genes are, you can see on the left here, this is a youthful pattern of gene expression, and over time, for reasons that we now essentially understand pretty well, some genes get switched off, some genes get switched on, and we lose the recipe. Well, no, take that back. We don't lose the recipe. The recipe's intact. It's the chef that loses the ability to read the recipe, so that what would be great is if we could reverse that and allow the cell to read the right genes at the right time, instead of having cells that, we're finding in a mouse that in an old mouse, a liver gene is being expressed in the brain, as an example. That we've done the whole genome, and we see that this is a fundamental process of aging. It's really emerging. You may not have even come across this idea. The old idea of mutations causing aging is essentially now falling away, and this is overtaking it as the major theory. Now, hopefully this movie will play for you. Nope, it's gone dark. Is it loading? Of course, it played beautifully before. Guys, it's gone dark. Are we done? All right. You know what? I'm going to actually just use my arms. Okay, this is the movie. What it was just an animation of diving through the brain into a cell, into the nucleus. It's showing up, starting on this screen a bit. Do you see it? There we go. All right. So that's the chromosome. Chromosomes are, of course, made of protein and DNA. DNA is wound up around those pink balls called histones. And on those histones, the revolution has been to realize that the chemical modifications on histones control which genes are on and off, which genes or which parts of the genome are susceptible to breaking. And what we've discovered is that there are a set of enzymes that modify those proteins to control this process. And these are called chromatin modifying proteins. And a major set of proteins that we've discovered, there are 7 of these in our bodies, are called sirtuins. And those seem to control the aging process. Now, we can speed this change up, actually. We originally, as I said, discovered this in yeast cells. We could speed up aging in the yeast cell by messing up their chromatin, as it's called. But we've been working really since 1999 to test this model in a mouse. If we mess up in a semi-natural way how the DNA is packaged in a nucleus, in a mouse, what happens? Now, my colleagues in my lab, we took bets. What happens if you do this in an animal, mess up with that? And actually, the way we do it, if you're wondering, is we create a few DNA breaks. It's a bit like having very defined X-rays, but they don't cause mutations. They just cut with a gene from a slime mold, an enzyme from a slime mold, and the cell repairs it. But that process of having to reorganize the nucleus and unpack the DNA and repackage it over a period of 3 weeks in this mouse experiment leads to this change in the organization of how the nucleus is packaged. So my colleague said, okay, the mouse is going to die. About 30% said that. Another 30% said nothing's going to happen. And another 30% said they're going to get cancer because you're cutting the DNA. And whatever's left, 10% said you might get aging. And that would be the home run, right, if we could actually do that. Okay, so this is the control mouse. This is the mouse that has not been exposed to this epigenetic change, as we call it. We call these mice the ICE mice. We haven't published them yet. You're one of the first groups to ever see this. ICE stands for inducible changes in epigenetics. This is a littermate that does not have these changes. Okay, it's typical black 6 mouse. And they are at the age, in this experiment, around a year. Okay, so a mouse at 1 year is not too old. It's like a 40-year-old. Okay, but what I've got here for you is the twin of this mouse, essentially genetically identical except for our manipulation. Hopefully she's going to come in. You going to play? Oh, come on. Do I have to wait? Or do I have to pretend I'm a mouse? Can you see that? Yeah. And there's another one. And they're all the same age, and they're genetically identical. They don't have mutations, but they have had their epigenome, the packaging, altered in a way we think is reminiscent, if not very, mimicking normal aging. And okay, big deal, you get a grey-haired mouse. What's so funny about that? Well, we've analyzed these mice for 3 years now, and every aspect of aging in a human is mimicked in these mice that we can look at. If you want to just throw a list of diseases at me, we can tick them off. From memory loss, diabetes, it mimics osteoporosis, skin aging, alopecia. I think that this mouse is going to be a very useful model for medical research. And if you have a model of Alzheimer's disease that does not fully mimic the disease, for instance, you don't have neuronal loss, that's one of the problems with mice. How do you age a mouse 80 years? You can do it this way. So I think what we'd like to try, and we would love to collaborate, to look at this mouse as a way to age the mouse model on top of, say, typical genes like presenilin 1 or A-beta. So here's our favorite sirtuin gene. I mentioned we discovered these in yeast 20 years ago. There are 7 of these. This is number 1. And this is the human ortholog of the yeast gene that we discovered. And so it's a blue dot here, but it's actually an enzyme, as I mentioned, that controls chromatin. And what we've discovered is that these genes respond to dieting and exercise. We believe they explain why caloric restriction extends lifespan in many different organisms. And we also believe, and have a lot of evidence as a field, that these genes are the reason that exercise and being lean is good for you, okay? They may not be the whole reason, but it's a different way of thinking, right? That it's a genetic response to adversity, and your body thinks it's under threat for survival, and it turns on these defensive genes, these sirtuins. They impact other pathways as well. If you look into the aging literature, you'll find mTOR and AMP kinase, and these are central to that network. What we've been trying to do for the last decade is trying to find small molecules, safe molecules that could be given to not just people in their midlife, but true disease patients, Alzheimer's patients, diabetics. We find these molecules that we're developing to be highly anti-inflammatory as well. And so let me tell you about those. But as you can see from this diagram, they're broadly protective of the body. If you're wondering, are we just going to talk about mice the whole time? Well, no. A couple of facts. One is that there are variants of SIRT1 in humans that have been shown to predict mortality. And the other thing is that we've done some clinical trials already in humans. Not on the brain, unfortunately. I don't have that data for you, but they are safe and anti-inflammatory in humans. All right. So this one's a summary slide. It's from a review from 2012 that shows you the roles of SIRT1 in brain diseases. So we've got, actually, particularly Alzheimer's disease, sorry. So we've got this gene controlling a number of different processes. I said that it controls the nucleus, but it's also controlling other proteins as well. Proteins that include, you can see up the top, there's tau phosphorylation is removed, or acetylation of PGC1 alpha that controls the energy in mitochondria. And at the bottom, we've got anti-inflammatory action. So we think that a drug that would safely get into the brain and work on SIRT1 could be very useful for treating Alzheimer's disease. And one of the molecules, I want to mention this now because I might forget, one of the molecules that we first discovered back in 2003 is called resveratrol. It's from red wine, it got all that hype. But resveratrol actually was in a human clinical trial the last few years, and there is some published data that it does slow Alzheimer's disease in a cohort of about 100 patients. But we're looking for things that are better than resveratrol right now, and I'll tell you about those at the end. So evidence that these genes control memory is the following. Hopefully this video will play too. So the setup is you can see mutants versus normal wild-type mice. And these mutants are lacking the SIRT3 gene, not the SIRT1, but SIRT3 gene. That makes an enzyme that's in mitochondria, and we have found that this protein controls the release of substances from mitochondria in response to challenge with A-beta. And though this isn't a memory test, I do have that data, but I thought this one I'd show you. This one is partly a memory test, but also partly a skill test. What we do is we put them on this rotating rod and we ask how quickly can they learn that they have to walk on this rod to stay on. We've got, for the aficionados, we've done Morris water maze and fear conditioning as well, and they're terrible at it. Is she going to play? Is it saying not going to play? All right, so what you'd see, what you'd see is that the mice, the mutants, just fall off. They just can't learn anything. They're completely stupid. Conversely, if we activate this gene, the mice learn better. And so we think that this is also pretty interesting. Now, genetic manipulation is not going to happen in humans, at least not for the next few years. So we need a pharmaceutical. And so one of the problems with sirtuins is you need more activity. It's very easy to inhibit an enzyme. That's what the pharmaceutical industry was built on. But to inhibit them to activate them is something quite different. And there's really only a few examples in the whole of medical research where this has been done. But we found this resveratrol molecule in 2003, and it was activating. We couldn't convince ourselves that it wasn't. And so what this led to was a whole series of molecules that activate the SIRT1 enzyme and modify the chromatin and seem to reverse aspects of aging. So you can see resveratrol's up the top left. We're up to actually the 5th generation of molecule now that's going into humans again next year. And in full disclosure, I consult for GlaxoSmithKline, who run these programs now. So what do we know about how these molecules work? At the enzymological level, they actually bind to one of the arms of the protein, and the protein gives its protein target a bear hug, and without it, the arm is floppy. If you feed these molecules to mice, just put it in their diet, these are a couple of molecules in the green and the blue graphs, they live longer and they're healthier. Now these are early studies, of course, they're not living dramatically longer, but if this was a human population, I'll take an extra 5 years. And we know that this is through the SIRT1 pathway. The molecule called 2104, which is in the blue, was the one that went into the human studies, both orally and now we're going into topical for psoriasis. But we can do better than this, I believe. This is old generation technology. We can activate all the SIRT2 enzymes at once, and that's by changing the levels of NAD. What I haven't told you yet that's essential to know is that these enzymes sense the levels of NAD in the cell. You'll know NAD well. You would remember, at least from high school, that this is the molecule that cells use for redox reactions. What we've discovered in our field is that NAD levels aren't just for chemical reactions. They're also, NAD is used as a sensor in the body for stress and disease. And that as we get older, we lose 50% of our NAD levels. And in mice, if we boost the levels back up, let me show you and tell you what happens. So what we've been doing to raise NAD levels back up to youthful levels and activate these sirtuins is to give them precursor molecules so that the cells can make more NAD. And that's just shown in this slide very simplistically. NMN stands for nicotinamide mononucleotide. It's a relative of vitamin B3. It's fairly low tech, but it seems to do remarkable things. It does boost memory in mice. It does protect them from cancer. And now I'm just going to cross my fingers that this video works. These are two mice, one of which has been drinking NMN and the other one has not for a couple of months. And hopefully you can tell the difference. The ones on NMN actually can go, in this experiment, 60%, 59% longer without getting tired. And we've traced this to the mitochondrial boosting and energy production, as well as increased capillaries in the muscle. This has, is he? Oops. Do you know, I've never seen that. I haven't seen that before. Usually I flick fast. Okay. All right. And so what we did was, so these were elderly mice, I forgot to tell you. And what we published, now going back about 18 months ago, was the fact that we could take old mice that were 2 years old, give them NMN for just 1 week in that experiment by IP injection, and reverse every aspect of aging in their muscle, ostensibly by activating these defenses against aging through sirtuin activation. And so the media was excited about that, reversing aging, not as crazy as you think, and hopefully I've convinced you that it isn't crazy to try this research. And even though we're still at the level of the Wright brothers compared to a Concorde, at least we know we can fly. And so I just want to end by saying that another reason to do these studies is for families. You know, my mother here, who was sick and made me go into medical research to do this. You know, this is a picture from a couple of years ago, but she passed away last year. When people say to me, "Oh, aging studies, you don't play God, blah, blah, blah," I say, well, do you want your parents around for another couple of years? Would you like them not to suffer in the last few years of life? Absolutely. Okay, then, shut up. Okay. So I'll end there, and we'll take questions later. Thank you very much.